Welcome to Notes from the Lab, an ongoing series of conversations on the past, present, and future of type 1 diabetes, and of course, the BCG vaccine. My name is Denise Faustman. I'm head of the immunobiology labs here at Mass General Hospital and Harvard in Boston. And my guest today is Dr. Alexander Fleming, but if you know him and you're a friend of his, you're going to call Zan. So Zan is the founder and executive chairman of Kinexum, a regulatory consulting firm that actually does a lot of work in type 1 diabetes. So welcome, Zan. And what I'd love to learn first is a little bit of your background and how you ended up on the regulatory side of getting products to the public. And it's my assumption your particular love of type 1 diabetes and getting drugs there. So please fill me in on your journey.
Well, thanks, Denise, and what a great honor to be with you. I've long admired your work. And of course, we have been at it in the type 1 diabetes space for decades. And I like to think that you and I march to a different drummer than the crowd. And we, in our own ways, have gone out to try to make a difference by following the evidence and always with the patient and high unmet need in mind.
Mm-hmm.
So great to be with you. And just to give you a little bit of background, I am an endocrinologist by training.
Wow, I didn't even know that.
Long story short, I was asked by a former classmate who at that time was at FDA
Mm-hmm.
if I would come up to FDA to help out for a period of time. And this was sort of as things were revving up in the diabetes space, but still pre DCCT. This was in the uh, uh, late 80s.
Okay.
And so I was asked to come up, and it turned out that things were really heating up in the diabetes and endocrine space. And it turned out to be an exhilarating opportunity to make a difference. I was there when we approved, for example, metformin. And at that
Oh,
time,
wow.
it was a very
Wow.
controversial decision. You might not believe it, but it was opposed by a number of experts who said we should never have
So
a
you're
bike
used to
one.
this. You're used to
Yeah,
this theme. It's wrong. It's wrong. It's wrong. It's wrong.
yeah. So that was just one example. We had the first GLP-1 agonist, and
Mm-hmm.
we had a number of new technological advances, recombinant insulin, which was, I mean, we're all just used to insulin being manufactured, but some people may not know it used to come only from animals.
Yeah.
And that transition occurred during my career. So
Wow. Wow.
I was just right in the center of action of therapeutic development for
Mm-hmm.
diabetes. And it was during this time at FDA that I developed a particular appreciation for type 1 diabetes
Mm-hmm.
as being the area where there is highest unmet need
Mm-hmm.
in not just the diabetes space, but I would say the endocrine space.
Mm-hmm.
And so from that time, like you, I've had a passion for seeking treatments for type 1 diabetes, disease modifying treatments, not
Mm-hmm.
just insulin or insulin pumps. And I'm very interested, involved in technology as well, insulin pumps and glucose monitoring devices and so on.
Mm-hmm.
But my passion has been type 1 diabetes. And it has, along the way, created great relationships and friendships, enduring friendships
Mm-hmm.
along the way. You're being an example. But for example, Jay Schuyler, another preeminent expert in the field, and Chantal Mature and I recently published what I think is a very important article in Diabetes Care, a leading diabetes journal, on the importance of C-peptide as a regulatory endpoint for approving disease-modifying therapies for type 1 diabetes. We might come back to that later, but those are examples of the kind of friendships and collaborations that
Developed
been
from these things.
developed out of this. It
Yeah.
was sheer dumb luck. Well, as you know, what does matter and what patients, and
Hmm.
I mean to say people with type 1 diabetes want is not just a life of unhindered um,
Hmm.
lifestyle and diet. And I mean, they want to be free of technology.
Yeah.
They want disease modification.
Hmm.
They don't want to have to wear pumps and continuous glucose monitors.
Yeah,
I mean, they're
absolutely.
certainly doing it, and they should be doing it today.
Hmm.
But what you and I are working for are what I would call practical cures,
Hmm. Hmm.
ways of making people more or less free of technology and able to live a normal life and without fear of hypoglycemia
Yeah.
or
So, don't
complications.
you think that people with diabetes have been kind of forgotten? I mean, it's
I
kind of
do.
like be happy with these devices, be happy with this insulin. We're going to give you a new insulin pump next year. And
Yeah,
it's kind of like, where's the immunotherapy? Where's the next big step for me? Not me, but people with this disease. It's
yeah,
kind of been a chase only after the not yet diabetic kids, but people with the disease are like waiting too for new innovations as well.
for sure. And again, we see the great value of
Hmm.
technology for
Hmm.
managing diabetes. It is
Hmm.
making life better, and it is improving clinical outcomes for people with type 1 diabetes.
Hmm.
But it's again, it's not where we want to be.
Hmm.
We're
Hmm.
still reaching for the stars. It really comes down to those few cells in the pancreas that secrete insulin
Hmm.
and the autoimmune process. You would have thought we could have worked it out and cured it,
Hmm.
been able to prevent it
Hmm.
uh, years ago. And it's not for want of trying.
Hmm.
It has been intensively worked on um, by people just like yourself.
Hmm.
And yet, it's just turned out to be scientifically a number of big challenges. First of all, safely being able to alter
Hmm.
the autoimmune process. And this is where you're an expert.
Yeah, yeah.
Um, uh, then, just as daunting is being able to get these cells to grow back. That
Hmm.
is, to return after being destroyed by
Hmm.
autoimmunity. And this is what we need for the great majority of people who have type 1 diabetes, who've lost almost all of their insulin secreting beta cells. And they cannot benefit from a autoimmune treatment alone. They
Hmm.
may need that. But what they also have to have, if they're going to restore normality,
Hmm.
is a way of growing these cells back. And you
Hmm.
would think we would have been able to figure that out.
Hmm.
We're able to manipulate cells with CRISPR technology and
Hmm.
all kinds of other highfalutin genetic approaches. We're able to even make insulin secreting cells in the laboratory or even in manufacturing facilities. But we have not been able to crack the nut of getting the body to grow back insulin secreting beta cells or islets.
Mm-hmm.
And that, I think, is fundamentally why progress has been very slow. And again,
Mm-hmm.
not for want of trying,
Yeah.
and not because there aren't a lot of smart people in the
Mm-hmm.
game, but it just has been tough, tougher than even.
Anybody thought. Yeah.
But what we now have are many treatments for controlling type 2 diabetes,
Mm-hmm.
glycemic control. Um, we have only one treatment for preventing or slowing the um, loss
Progression. Yeah.
of insulin
Mm-hmm.
secretion in type 1 patients, and that is teplizumab. And so there's a big, big difference in what we have for type 2 diabetes and type 1
Yeah,
diabetes.
for sure.
And of course, we have insulin, of course. We have increasingly more sophisticated kinds of insulins, longer
Mm-hmm.
acting insulins that
Mm-hmm.
make it somewhat easier or convenient for people to get good glycemic control, whether they're type 1 or type 2 people. But again, what you and I are interested in is disease modification,
Mm-hmm.
not just
Mm-hmm.
disease treatment.
Yeah. Yeah.
That is largely under control for type 2 diabetes.
Mm-hmm.
For
But
type
not
1,
type 1. I mean,
yeah,
treatment still is very
yeah.
difficult.
Now, I will say there is a challenge posed by the diabetes technology to
Mm-hmm.
developing disease modifying treatments for type 1 diabetes.
Mm-hmm. Mm-hmm.
Let me explain that. The great thing about closed loop systems, that is, an insulin pump paired with a continuous
Mm-hmm.
glucose
Mm-hmm.
monitor, is that they really do a great job of both improving glycemic control
Mm-hmm.
and reducing risk of hypoglycemia. And more people are using those closed loop
Mm-hmm.
systems. In fact,
Mm-hmm.
in all the Harvard system clinics, you won't find um, any clinician who is not urging their patients to use closed loop systems. And so when we put people in clinical trials of disease modifying type 1
Mm-hmm.
treatments, they're on these pumps and monitors.
Mm-hmm.
And it's hard to show a benefit on A1's hemoglobin A1C or in reducing risk of hypoglycemia.
Mm-hmm. Mm-hmm.
So, what is the only practical way of showing a benefit with these treatments, like with teplizumab?
Mm-hmm.
It's C-peptide.
Yeah.
And so that's why C-peptide more than ever is a very important endpoint. And it's really our only practical endpoint for being able to get a read on the value of a product for either reducing the progression towards type 1
Mm-hmm.
diabetes or slowing the loss of beta cells after type 1 diabetes
Mm-hmm.
is diagnosed. And so that's the good news and the bad news. The
All
good news
together.
is the
But
technology really does work. It
mm-hmm.
is helping people to live better. I mean, to manage their condition better and more conveniently, but it's also
I think
making it
about all the
harder.
moms with the CGM iPhones next to them at night. I mean, you
Yeah.
gotta admit, I mean, compared to waking up and hearing your kids seize in the
Oh,
middle of the
yeah.
night, I mean,
I mean,
that's changed their lives. But it's still, as you said, I call them gizmos. They're still gizmos. They're not more permanent solutions to blood sugar control.
they are not. They
Yeah.
simply are not.
I mean,
That's not what people want.
no, I mean, that's why, I mean, we're biased, right? But that's why we love BCG. I mean, it doesn't work on the pancreas. It doesn't give a damn if the pancreas has anything left in it. You can have diabetes 30 years, 20 years, and your hemoglobin A1Cs come way down and your insulin use comes down. And it's related to metabolism. I mean, it sounds more
Yeah.
like I'm a type 2 diabetes expert. I tried to avoid all those type 2 diabetes seminars. And keep in mind, here is all type 2 diabetes. You know, it's metabolism from John Potts and David Nathan and Joe Averick, some of these real pillars. And it's always been lectures on metabolism. I tried to ignore all of them, right? Because this is not what I'm interested in. And lo and behold, like 10 years ago, we realized type 1 was a metabolic disease. And I guess if you sit there long enough, you catch the bug and realize that there was this gap, talking about going in opposite direction, there was this huge defect in people with type 1 diabetes that was not pure autoimmune, not pure immune mediated, killing islets, but they weren't using sugar correctly. Talk about a 180. Okay. So, I mean, I'm kind of famous going this way when everybody else is going that way, but like you, we follow the data. We don't follow the dogma. And the data tells you where to go. So all I can say is I feel fortunate to have sat through so many grand rounds of metabolism to finally have my eyes open to see that, oh my God, there's this huge defect in metabolism that people missed somehow. So it will be very well complemented by something happening in the pancreas, right? And
Yeah.
so that's why we have all these new onset trials and pediatric trials going right now. We got 250 kids enrolled. Nobody drops out of these trials. I mean, these parents are in for it. You know, they
Yeah.
know their kids. I mean, the kids already have diabetes, right? Some of them have had it for 8, 10 years, like the adults, but we have
Yeah.
the new onsets as well. So it gives them another way to think about the disease to get better control, but they're all on all the closed loop systems, as you say. So we got to beat those every single time
Yeah.
to make the FDA
And that
happy.
it makes it hard. But
It makes it hard.
we should talk about a very important distinction which
Mm-hmm.
relates to unmet need.
Mm-hmm.
Um, as you know, most of the research or the development, the clinical trials
Mm-hmm.
for disease modifying treatments is focused on people with so-called new onset type 1 diabetes.
Mm-hmm. Mm-hmm.
And that is defined today as roughly anywhere from 30 to 100 days from
Mm-hmm.
diagnosis. In other words, that's a very small population.
Mm-hmm.
And
Mm-hmm.
in fact, it actually qualifies as an orphan population.
Because how many
It's
kids per year? Probably 30,000, 40,000.
roughly, yeah,
Yeah, fall in
30
that
and
category.
about this maybe 20,000 adults. So still an orphan population, well under
Mm-hmm.
the 200,000 patient prevalence number that FDA uses.
Mm-hmm.
And so it's a small number. And
Mm-hmm.
yeah, that's where we're pouring 90% of our effort with
Yeah.
treatments to try to halt
Mm-hmm.
the autoimmunity in people who still have some insulin
Mm-hmm.
secretion to protect.
Mm-hmm.
But who's left out? It's the 96%
9%
of
that have the
people
disease.
who have, and it's what we call established type 1
Mm-hmm.
diabetes.
Mm-hmm. Mm-hmm.
Very few companies are working on that.
Well, they think it's impossible, right? I
Right.
mean, I remember you can see my probably seat in the back here. There's a famous endocrinologist, no names mentioned to protect the innocent, sitting on that couch, somebody you know. And when we first started our phase 2 trial, it was kind of like, what the hell are you doing? You're going into people that have diabetes? Are you nuts? And
Yeah.
and I said, well, there's really two logistic problems. As you know, there's a lot of big companies and they're all fighting over the not yet diabetic kid. And they're all fighting over the recent onset, the
That's right.
30,000 or so. So we have no chance of ever getting sufficient enrollment in those. So let's just take that one off the panel. And then they said, well, then why are you going into people that, well, I said, well, need. And it's like, what's the science? And I said, well, old fashioned BCG is still, even today, the only immunotherapy that works in end stage mice. End stage, high blood sugars, 500 and 600, pancreas, a sea of white blood cells. Even CD3 only works in mice, in new onset mice. You go 3 weeks out, it doesn't work. So if we're getting this data in mice, not that most mice are men or vice versa, most men are not mice. If we're getting this data in that kind of end stage, we should start people in that end stage. So it
Yeah.
was really going into these very established diabetics
Yeah.
that we did financially because we couldn't afford to try to compete with Sanofi or Tolerax or any of those companies to recruit those kids. But also, we had the first mouse data saying there was hope. So that's how we got going with all these people with years of type 1 diabetes, years.
Yeah.
And I can tell you, many people came here to the Mass General for grand rounds. And they go like, what are you doing? Oh, we're going to do immunotherapy in people with diabetes. And it was just like, you could just see them turn off like that's an impossible possible goal. But it was really lack of money that drove us to
Yeah.
that, you know? And
Well,
so we've had great rewards going in that direction.
but that is the huge unexplored area.
Yeah.
That's where the greatest need is in
Mhm.
aggregate.
Yeah,
And
absolutely.
it's not to minimize the importance of helping children who've just been diagnosed with type
Hmm.
1 diabetes and
Yeah.
adults. And as you know, it's roughly half and half.
Hmm.
People who are diagnosed with diabetes, the mean
Hmm.
age is about 18. So
Hmm.
plenty of adults, even in their 60s or 70s,
Hmm.
come down with type 2
Autoimmunity.
diabetes.
Yeah.
But as you say, it's the younger children who are being sought after by the companies
Yeah.
because they have a much faster rate of
Yeah.
decline.
The trials become a lot longer.
Yeah.
Yeah.
But what you are doing is great in that you're going after a much larger population.
Hmm.
And whether you have the cure or not, at least if you can help with improving glycemic control, that's a help.
Yeah.
And if you can improve glycemic control, that indirectly will reduce the risk of hypoglycemia. It will
Hmm.
reduce the need
That's
for.
also important because you can always give more insulin. Everybody knows that
Exactly.
point.
Well, and Denise, as you know, insulin is the most dangerous drug we use on an everyday basis.
Yeah.
It has the least margin for error,
Hmm.
what we call the lowest therapeutic index.
Hmm.
And people with type 1 diabetes in particular know this. They have to fine tune their insulin
Hmm.
dose. Unlike people with type 2 diabetes, it's not nearly as challenging.
Yeah.
And of course, again, the technology has helped. But the fact is, insulin, as we inject it into the body,
Hmm.
is not an easy proposition.
Hmm.
It either requires a lot of attention and or
And
a lot of
monitoring.
technology.
Hmm. Hmm. And that's where we are today. So if you were to go back 20 years and start up a research lab, I'm going to put you back in a lab. What project would you work on as it
Well,
relates to type 1 diabetes? What would you
I,
go after?
I would have to admit that I am a dreamer about restoring insulin secreting cells,
Hmm.
what we call islet neogenesis or beta
Hmm.
cell um, replacement, or not beta cell regeneration.
Yeah.
Um, as you know, when women with type 1 diabetes become pregnant,
Mm-hmm.
their insulin requirements actually drop.
Mm-hmm.
And this is evidence for some kind of return of insulin secretion, along with maybe some immune changes. But by and large, I think this is telling us that even adults had the capacity to bring back some of their beta cells. And you were very important in showing that people like we call them the Joslin medalists, have
Yeah.
some residual insulin secretion, even after 50 years of having type 1 diabetes. So there again, that brings me back to my passion of wanting to find ways of hormonally inducing beta cells to grow or
Mm-hmm.
islets to grow. And so that's been a project of Susan Bonner Weir and her husband, who unfortunately recently
Yeah,
died.
just passed. Yeah. Yeah.
Um, but the point is that's a even less active field right now than
Mm-hmm.
I believe it should be. And so that brings us back to the present. And
Mm-hmm.
I think we can talk about BCG because I know you only have a few minutes. Um, you presented some very exciting data in New Orleans at the American
Mm-hmm.
Diabetes
Mm-hmm.
Association meeting. And I think the data are very encouraging. And again, it's because it provides a benefit to the big majority of people with type 1 diabetes, people
Mm-hmm.
with established type 1 diabetes.
Mm-hmm.
It does result in an improvement in glycemic control
Mm-hmm.
as reflected by hemoglobin A1C. And that's an approvable benefit.
Mm-hmm.
And so we know a lot about BCG. It's used globally
Mm-hmm.
as an effective vaccine for tuberculosis. We don't use it in the US for complicated reasons. But it's something we know a lot about. It's safe. It could easily be used by people with type 1 diabetes to give them a little edge, to give them improved glycemic control, and not to have to worry about off-target effects, that is toxicity, side effects. So I think it's exciting work. And again, it reflects
It's a long slog to get there.
long
But
slog
you
to get.
know, it's actually our sixth clinical trial, and some of those trials along the way were open label because we just had to
Yeah,
test it again and again. But you know, when you bring hemoglobin A1C with people on closed loops down 15%, and then when you look at their CGM, and it's not time and range anymore, it's time and normal glycemia. Okay. And so that means it's tight knit. You know, they are no longer doing this like more insulin. They are that. And by year 8, 41% of the people have a normal hemoglobin A1C. Normal. They're on insulin. They're on insulin, but usually long acting insulin. So that helps people out. That helps
for
people out.
sure, for
And
sure.
it's totally safe. And so after 3 billion global doses, we kind of have that one nailed.
Yeah,
Its disadvantage is it takes a while to kick in. So it's not like you give it and next week you're at your endocrinologist with a better hemoglobin A1C. It takes a couple years, but the
yeah,
durability, it's to me a no-brainer of
yeah.
helping people out. Yeah.
Well, that's right. And it's like any other drug that we use to control metabolic disease. If you take
Mm-hmm.
a statin, you're
Yeah.
not going to feel any benefits. You're not going to even see benefits
Mm-hmm.
in a controlled
Mm-hmm.
trial for a few years where you
Yeah.
start to see the curves diverge.
Yeah.
So you just need to know that you're going to benefit over the longer haul
Hmm.
and that the benefit to risk relationship is positive. And in
Hmm.
this case, there's virtually no risk.
Yeah, yeah, absolutely. So we're excited about it. I think all those long-term diabetics that were skeptical can now feel a little better that if we can get that out there, what a world of difference. And nobody else, as you mentioned, working in that area. So it's totally
Yeah,
open. We're not
I,
competing for patients.
yeah, I think you had the feel
Kind of
pretty
locked
sewn
up.
up.
Yeah, yeah, yeah. But that's what we're doing every day. And, I really appreciate today taking the time to discuss type 1 diabetes. It's certainly an interesting field. I think the people in type 2 are more boring. The type 1 diabetic community has much stronger opinions. But I think inch by inch, the only way to prove these things is randomized controlled trials. And certainly your expertise in that is what helps all of us to get through the hurdles to make sure everybody believes the data. So I can't thank you enough for your expertise in regulatory medicine and hurdles, and also just your perspective on things that I didn't know about you and also didn't know about the diabetes field. So this has been a true pleasure today, and we really thank you for contributing this
The
podcast.
pleasure and honor is mine.